To draw a single breath, the human body must expose roughly 75 square meters of internal surface area to a dry, pathogen-rich external environment. That creates a massive physiological paradox. To allow for passive gas diffusion into the bloodstream, the tissue barrier exposed to that outside world must remain incredibly thin. This diagram of the gross anatomy shows the respiratory system dividing the workload. It splits into a conducting portion up top to clean incoming air and a respiratory portion deep inside for gas exchange. This upper pathway safely transports air to these microscopic terminal ends. The respiratory tract manages this contradiction through a strict histological gradient. As the airway branches deeper into the lungs, it systematically strips away its own structural defenses. Every epithelial cell and connective tissue layer we observe along this path is specifically engineered to solve a distinct physical problem dictated by its location in the airway. We start high up in the trachea and primary bronchi. Down here, the primary objective is keeping the massive airway open and trapping large external debris. Looking at this cross-section of the tracheal wall, you can see continuous C-shaped rings of hyaline cartilage. These provide the rigid structural support required to prevent the airway from collapsing under negative inhalation pressure. At the cellular level, this pseudostratified ciliated columnar epithelium sits on a thick blue basement membrane with a lush cover of apical cilia. Interspersed goblet cells secrete mucin directly onto the surface to trap airborne particles, acting as the mucociliary escalator. The second step relies on those columnar cells. Each one uses over 250 apical cilia beating synchronously to continuously propel the trapped debris upward toward the pharynx for elimination. When this system faces chronic exposure to environmental toxins, like heavy cigarette smoking, those cilia become immobilized and the clearance mechanism halts entirely. The consequence is squamous metaplasia. To survive the constant irritation, the highly specialized respiratory epithelium reverts to a simple stratified squamous tissue, producing a precancerous cellular dysplasia. The upper airway's thick, heavily fortified architecture prioritizes maximum physical defense and structural rigidity at the complete expense of gas exchange. Moving deeper into the lung, past the 10th generation of branching, the pathways shrink to less than a single millimeter in diameter. These are the bronchioles. In this bronchial cross-section, the architecture simplifies abruptly. Cartilage plates disappear entirely, and the epithelium drops to simple ciliated columnar or cuboidal tissue. With cartilage gone, structural control shifts to a dense circular layer of smooth muscle wrapping the lumen. Relying on muscle creates a specific vulnerability. In conditions like asthma, antigen-triggered mast cell degranulation causes severe bronchospasms. This specific muscle layer violently constricts, dangerously narrowing the airway. As the airways narrow further into terminal bronchioles, goblet cells and mucus must vanish. If they remained, the thick fluid would physically plug the tiny tubes. In this high magnification section, the cuboidal epithelium introduces a new defensive strategy: non-ciliated exocrine bronchiolar cells, or club cells. Notice the distinct dome-shaped apical ends of these cells. They are packed with secretory granules pushing right up against the luminal space. These cells shift the defense from physical to chemical. They detoxify inhaled xenobiotic compounds using smooth P450 enzymes, and they secrete antimicrobial peptides and cytokines into the local environment. The bronchiole is a vital transition point. It trades rigid cartilage for dynamic muscle control and swaps physical mucus trapping for localized enzymatic defense. Finally, the pathways open into the deepest respiratory portion of the tract: the respiratory bronchioles, alveolar ducts, and alveolar sacs. Here, the exclusive physiological goal is pure gas exchange. This scanning electron micrograph reveals the sheer scale of the exchange zone. An adult lung contains roughly 200 million sac-like alveoli interconnected by tiny alveolar pores. 95% of this inner alveolar surface is lined by type 1 alveolar cells, also known as type 1 pneumocytes. To facilitate diffusion, these cells are heavily attenuated. Their organelles are tightly clustered around the nucleus, allowing the remaining cytoplasm to stretch to a thickness of just 25 nanometers. This ultra-high magnification TEM reveals the lung's ultimate engineering marvel, the blood-air barrier. It separates alveolar air from the capillary bloodstream using three exact components: the incredibly thin type 1 cell, the capillary endothelial cell, and their completely fused basal laminae sandwiched between them. By fusing those basement membranes together, the system compresses the total physical barrier thickness to an astonishing 0.1 to 1.5 micrometers. This radical vulnerability, placing dirty atmospheric air mere fractions of a micrometer from circulating blood, is the absolute structural requirement to optimize Fick's law for rapid passive gas diffusion. But this microscopic architecture introduces a new problem. The alveoli must be coated in a thin layer of fluid. Inside a sphere just 200 micrometers wide, the surface tension of that fluid vectors sharply inward, generating immense contractile force. Without a biological intervention, this physical tension would cause the delicate alveoli to collapse entirely upon every exhalation. The solution is the type 2 pneumocyte, seen here bulging into the airspace with small apical microvilli. It synthesizes a mixture of lipids and proteins, primarily DPPC, and packages them into these dark, specialized granules called lamellar bodies. The continuous exocytosis of these lamellar bodies creates a surfactant film over the aqueous layer. This drastically lowers the fluid surface tension, maintaining alveolar patency with far less inspiratory force. When premature infants are born before their type 2 cells have fully differentiated, they suffer a critical deficit of lamellar bodies and surfactant. Infant respiratory distress syndrome illustrates the exact physiological stakes. Macroscopic airway patency and the simple ability to draw a breath ultimately depend entirely on microscopic lipid chemistry. The respiratory system is a masterclass in biological compromise. It cannot rely on a single uniform tissue type to accomplish its opposing goals. It begins by employing thick, rigid cartilage, viscous mucus, and millions of beating cilia to actively warm, filter, and physically defend the upper tract. In the middle airways, it discards that structural rigidity for dynamic smooth muscle flow control and deploys club cells for localized enzymatic detoxification. This diagram compares that thick, ciliated fortress against the radical vulnerability of the final destination, where all structural bulk is stripped away to optimize diffusion, and only the continuous chemical application of lipid surfactants keeps this ultra-thin, unprotected architecture from collapsing under its own physics. Human respiration is only possible because the anatomical airway meticulously and systematically sheds its own defenses, exactly at the microscopic point where gas diffusion becomes paramount.